So our second panel today is intended to be perspectives of some of the governments and some of the government entities that are involved in genetic testing issues. I will say this is one of my failures in organizing the conference, in that although I'm very, very happy with the three people we have, they're not the failures. I wasn't able to get anybody to talk about CLIA, the Clinical Laboratory Improvement Amendments Act, which plays an important regulatory role, which unfortunately there's not a CLIA agency exactly. Responsibility for CLIA is shared in many respects. So I hope that we will remember as we go through this that there is another important regulatory player in here, CLIA, which various people here on the panel and in the room can say something too. But I'm very happy that we have the three speakers we do. We'll start with Pamela Bradley, who is a staff fellow at FDA. Alberto Gutierrez, the head of the Office of In Vitro Diagnostics, which has been doing the LTDs, said, well, you know, I could come and talk, but you really want Pamela because she's the one who actually understands this. I've asked her to talk some about the LDT draft guidance, but also a little more generally, to the extent she can, about issues of genetic testing going forward.
Hey, Hank, can we flip? Can I go before Pam?
You want to go first?
Mm-hmm.
Gosh, spontaneity?
Yeah, well, we talked about it.
Okay. So our first speaker will now be Kathy Hudson, Deputy Director of NIH for Science, Policy, and other good stuff. And, you know, since she's paying for a fairly large chunk of Stanford, since NIH pays for a fairly large chunk of Stanford University, if she wants to go first, who am I to disagree? The great thing about Kathy, from my perspective, is not only does she represent the federal government's predominant biological research arm's perspective on this, but I want to claim her as a law and biosciences or biosciences and society person. Before she was at NIH, she was the founding director of the Genetics and Public Policy Center and did spectacular work there for many years, trying to figure out, among other things, what do people actually think and what about genetic testing. Our third speaker, I'm really pleased to have here because Stephanie Schulman represents the New York State Department of Public Health. We focus so much on the federal government that we forget that there are actually 50 states out there, plus a few other miscellaneous state-like jurisdictions, and they all have public health departments, and they all play a role in clinical testing, including genetic testing. New York is, I would say, the one state whose public health department has really taken seriously the issue of regulating genetic testing. So when we think about regulation of genetic testing, we're not just thinking about the federal level, but we need to be thinking about the state level as well. So we'll start with Kathy, then go to Pam, and end with Stephanie, and have lots of time for discussion. And this time I may even let the panelists discuss among themselves, as well as answer my questions and your questions. Kathy.
So thanks for indulging me to go first. I want to see if I can pull up my slides. Thanks, Hank, for inviting me to be here. It's nice to be on the West Coast. I am insanely jealous of Mark from the National Institute of Standards and Technology that has a West Coast branch. I think that's a brilliant idea, and I'm going to be the founding employee of NIH West. And I hope, I'm sure you will.
We welcome partnership.
I'm going to talk a little bit about sort of the broader perspective of genetics and genetic testing, and hopefully provide a good introduction to what Pamela will be talking about. As you all know, the NIH is responsible for investing in basic biomedical research and discovery, and certainly the discovery of the molecular basis of human diseases and disorders has gone up steeply. And Hank mentioned that before I went to NIH, or back to NIH in 2009, I had been running a little genetics policy think tank, which I started in 2001. But actually, I had started at NIH previously in 1995, working at the National Human Genome Research Institute with Francis Collins. We're sort of on round two working together. And certainly the investments that we have made in genetics research have paid off handsomely. So over the course of my professional career in genetics, we've seen an increase in number, complexity, and availability of molecular diagnostics. And also, impressively, an increase in the clinical relevance of this information. And you heard quite a bit about this from the last panel, so I won't dwell on this. Amazingly, the cost of sequencing genomes has dropped precipitously, and people like to refer to it as being faster than Moore's Law. And I try to say, at least for most lay audiences, certainly lay audiences outside of Palo Alto, they may not know what Moore's Law is, but I'll show you an interesting quote at the end of people who do understand what Moore's Law is. We have been involved and engaged with FDA intimately ever since Peggy became the FDA commissioner and Francis the NIH director about 5 years ago, and they actually published a paper together early on in their tenure about the need for coherent oversight and predictable oversight of molecular genetic tests. And then recently, with the FDA clearance of a sequencing platform, they again wrote an article together talking about that. And Francis tweeted about it, and if you guys don't follow him, you might sometimes tweet some pretty interesting stuff. So now we have one cleared platform for DNA sequencing, Illumina's MiSeq, and it was cleared without a specific intended use. And I thought that that was particularly important. And at the same time, they cleared 2 panels with cystic fibrosis mutations that were for use on the Illumina platform. So I think that paves the road for additional sequencing platforms being cleared by FDA and also for new panels of mutations. And Pamela, I think, will probably talk more about how that might work. Our role at NIH is to support the research that is necessary in order to uncover the molecular basis of human disease and to support innovation in technology and test development and to help evaluate the safety and efficacy of molecular diagnostics. Since we came back to NIH, we helped, Francis and I helped launch the genetic testing registry, which I'll say a word about, in February of 2012, and then built alongside the clinical genetic testing registry are a number of resources through the National Library of Medicine, many of which you are familiar with, but most notably ClinVar and Clinical Genome Resource, or ClinGen, and I'll talk a little bit about them. So the genetic testing registry, if you're not aware, is a voluntary genetic testing registry, which is available on the NIH website. It currently has, I think maybe I have the number on the next slide. Yeah, it currently has 23,586 tests that have been voluntarily submitted onto the registry. They include a large number of pharmacogenetic tests, as well as many other kinds of tests. Presently, GTR does not take tests for infectious agents, but may potentially expand into that arena. So the tests that are available or whose information is disclosed on the genetic testing registry, it's all completely voluntary and it is not validated by the NIH. There was some anxiety when we set this up that it wasn't going to be validated by us, but it has worked out fairly well so far. And they, here is sort of a breakdown of the kinds of information across analytic and clinical validity and clinical utility that is available for those different tests. And you can see a dramatic decrease in the amount of information or the number of tests which have associated clinical utility information. So the other resource that is somewhat newer is ClinVar, also a publicly available resource. And it is an archive of assertions that have been made about the clinical significance of genetic variants. And again, it's not validated by the NIH. And every time we do this, we have the same angst and long discussions with the attorneys about what does it mean and are we really giving the imprimatur of NIH by hosting such resources. But again, I think that the value is of value even without an expensive validation process. Presently, there are 98,000 variations that have assertions about clinical significance that have been registered on ClinVar, and again, it's publicly available. The data in ClinVar is fairly intuitive to be able to navigate, and it is also given a status. And so up in the top here, you see that this has 2 yellow stars, sort of like a restaurant rating, and it basically is how much evidence is there to support the assertions, clinical assertions about those variants. And in this case, there are 2 entries about this particular variant, and therefore it gets 2 stars. If there were a review by an expert panel, it would get a 3-star rating, and if there was a professional society or clinical guideline, there would be a 4-star rating. So this is, again, out there to help the community and be able to to curate some of the information that's accumulating from labs across the country. The other resource that we have started is ClinGen, which is intended to provide a resource of clinically annotated genes and variants to improve our understanding. And the goals are quite ambitious. First, to standardize the clinical assessment of variants and their deposition into ClinVar, to develop a consensus process for identifying clinically relevant variants, to curate genes and variants with multiple clinical domains, to develop some machine learning algorithms to improve accuracy and throughput for variation interpretation, and to disseminate and explore integration within EHRs. This is a big, sort of audacious set of goals, and this program is in its infancy, but we have very high hopes for this project, particularly as we're entering a stage where the need for more rapid review and assessment of clinical validity is really going to be pressing, particularly as FDA moves forward. So as we move forward, we need additional research to expand the evidence base. We need to understand the clinical relevance on the ground in real life of genetic variants. We need a nimble and risk-based regulatory system, which again, Pamela will talk more about. We need to promote broad access as appropriate, integrate clinical genetics into electronic medical records, which is a gnarly problem, and to create information resources for patients and providers. And importantly, and we haven't really talked today, and the other federal agency that's not here is CMS from their payers' perspective, is we need to establish clear policies for coverage of molecular diagnostics. So going back in time, when in 2000, President Clinton talked about the significance of the Human Genome Project, and he said, "I thought it would probably be the most important scientific discovery of the 20th century. I thought it held the promise of giving mothers a little gene card to bring home from the hospital with their newborns, which would tell them what their genetic profile is and give them a way of minimizing the risks and maximizing the strengths of their newborn babies." Of course, that was 14 years ago, and we're only now really looking at what does it mean to provide exome sequence information with newborn testing. But what's more interesting, at least to me, is a statement that the current president made on October 9th, just a few days ago, in a speech at an innovation meeting in Los Angeles, in which he said, "What's really going to be interesting over the next decade, 2 decades, is precision medicine or personalized medicine. Because of the work that's being done on the human genome and the ability to sequence your genetic makeup, and the costs are going down, actually faster than Moore's Law. They're plummeting so that pretty soon, for $100 or less, you'll get your entire human genome sequenced. And what that means, at a minimum, is you're going to know there's a certain diseases that you may be more prone to, for example, Alzheimer's," and he talks about how that's going to be a really great thing. He goes on to say, "Now, we're going to have to change how we regulate some of this stuff. We don't want bad information going out." Again, Pamela will talk about that. "We want to make sure that there's not a lot of hucksterism in this whole process. We are still going to need doctors and hospitals to make sure what you're thinking about actual interventions, like taking a drug. But the potential of this to really change how people think about their own health care is tremendous." And he goes on to talk about how he has put together a group to think about and usher in a new age of precision medicine. And dare I say it, I think that his activities and interest in this area may be one of the things that he hopes to leave behind as one of his legacies, and certainly is one of his scientific priorities. So watch this space. Thank you.
Okay, don't like following quotes from the president, but I will. Okay, I'm going to take us back to kind of the central question of how should we regulate genetic tests in the U.S. and tell you what FDA is doing in this area so that you have a sort of launching board for the rest of the day's discussion. Briefly going to talk about FDA authority and oversight, moving into the genetic tests that we already do regulate before the big question of LDTs and the framework. Okay, so in 1976, the Medical Device Amendment Acts, Medical Device Amendments to the Federal Food, Drug, and Cosmetic Act gave us the authority at FDA to regulate medical devices and then defined in vitro diagnostic tests, these laboratory tests, as devices. So this is under our purview, whether they are made in a lab or made in a commercial, by a commercial entity and distributed. We're charged with ensuring that these tests are safe and effective, that they do what they say they'll do, based on their intended use so that patients are not unnecessarily harmed. We have a risk-based regulation at FDA. Medical devices, including IVDs, are regulated to the extent that is driven by the risk of their intended use. So how this test will be used is very important in our assessment of risk. This means that a tongue depressor or a toothbrush, which are medical devices, have a lower regulatory bar than something like an artificial heart. So that makes sense. But how does this translate into the IVD world? With tests, it's based on the consequences of the false result. So you can imagine that if you get a bad test result in HIV, it's much worse outcome than if you get a bad result on a pregnancy test. Maybe, for some people. Okay, and so we have 3 classification levels. Class 1 being, and I'm going to use these because I'll refer back to them when I talk about the LDT framework, but basically, class 1, very low risk, minimal controls, general controls as we call them. Class 2, they're going to be more complex, so there's going to be a higher regulatory bar with these tests. And then class 3 is the most complex, highest risk test that we see. Again, based on the consequence of a false result, that would be what we look at. So just want to plug the FDA for what you get out of FDA oversight. It's an independent pre-market review. So before these tests come on the market at all, they've been looked at to make sure that the claims that they're making are valid. So the analytical and the clinical validity, as we've talked about, the data behind those claims is going to be evaluated by the reviewers. So that the test performance, the accuracy of that test, the predictive values of that test has been evaluated, and that we can have some assurance that what's in the label actually reflects what we know about that test. Had a lot of discussion about the clinical validation piece. That is something that FDA provides. We provide that assurance that those tests are providing the clinically meaningful results. And then there are a handful of post-market controls as well. So when things go wrong with those tests, we can help the manufacturers of these tests get it back on track and back into the market so that there's minimal harms to patients. So we have a fairly extensive experience with genetic tests. I know that the lore is that the vast majority of genetic tests are LDTs, and that's true, but the agency does have quite a bit of history with it. There's always the non-nucleic acid-based genetic testing that you might think of, but we seem to be more in the nucleic acid-based tests. We have them for cancer diagnostic purposes to assess the risk of recurrence of cancer, cystic fibrosis diagnosis, pharmacogenomics, heart transplantation. There's also a slew of microbial tests available for HPV, HCV, et cetera. The website up here has all of them listed. We try to keep it up to date. And then we also have a separate website for the companion diagnostic piece. And those are the tests that are required for the safe and effective use of the drug. They go hand in hand, and we have policies. We can discuss that later. I do want to go back to the nucleic acid-based test. Kathy mentioned this, and we did, I think, make very good inroads into providing a regulatory path for the Illumina, for next-gen sequencing in general, through the Illumina clearances last year. So we had 2 clearances related to the sequencing instrument itself, which provided the analytical validation. I think the methods that we went forward in getting this onto the market showed that the agency is willing to be flexible and to adapt to the technologies. There was no clinical validation needed because that didn't have a specific intended use. On the clinical side, there were 2 CF cystic fibrosis tests that were also cleared. And again, one of the most important things to that is that for the clinical validation piece, we allowed the test label to point to the Hopkins database on CFTR so that you would know from that database what's in there is a regulatory grade resource for understanding what those variants meant. And I think that's something that we think might be tenable going forward. Okay, but again, here we are, the vast majority of genetic tests are LDTs, and LDTs, as defined by the agency, are IVDs that are designed, manufactured, and used in a single laboratory, under a single CLIA laboratory certificate. And as you know, FDA has generally exercised enforcement discretion for LDTs. That's why we're having this discussion about who should regulate genetic tests and how they should be regulated. And just so that we're all on the same page, from the agency perspective, enforcement discretion is when we don't enforce some or all of the applicable regulatory requirements on a certain category of products, medical products, such as a drug or device. But the key point here is that just because we don't enforce our regulatory authority does not change the fact that the law does apply. So enforcement discretion is not unique to LDTs. The agency uses it for many other things. And there's different reasons for doing this. Sometimes it's risk, sometimes it's resources, sometimes things have changed and just for various reasons. So this happens, but we can also change. This is not a law, it's a policy of enforcement discretion. And I think we probably are all well versed in how the evolution of genetic testing, but LDTs in general, has changed over the past couple decades. Technology's changed dramatically, as we've seen. Marketing of these products has changed. The business models have changed. So more risk is associated with LDTs these days, in our opinion. And the consequences for this are that we have seen significant health adverse events, unnecessary healthcare costs of testing with faulty or unproven tests. And we're concerned that the lack of oversight in this area could undermine the progress in personalized medicine that we're seeing and that we all know depends on getting that right test result. Long history here, lots of recommendations along the way of having more oversight from the Secretariat Advisory Committees, from the NIH, from the IOM, tasking FDA with this. And we've tried in the past to do some sort of build control over the system with a piecemeal approach that really faced a lot of challenges along the way and heard very strongly from the stakeholders that they wanted to understand the broader LDT landscape and sort of have a feel for how they fit in. So don't carve off one part and only regulate that, like with the IVD-MIA issues, for those in the know. Obviously, we don't have time to delve into those, but so in 2010, there was a public meeting. We took back all of that public information that we heard, the comments, and worked very hard to balance all the differing opinions and concerns and with protecting public health to develop a framework. So the framework that was published as a draft guidance in the end of September and is now out for public comment is that. It's a draft. It's a proposal, and we really, I think the biggest message you can take away from me today is that we want to hear from everyone. We want to understand how it would impact you. What can we make better? What do you like? I will point you to the notice of availability, which usually people never pay attention to these things, but in it we have listed a fairly extensive list of things that we want to specifically hear about to make sure that we're saying, hey, these things are things that we might consider more conversation about. There will be a webinar for the people who really want to know the details on October 23rd. I'm going to provide now an overview of this framework, but I think, as Hank has mentioned, I don't think we want to delve too far into the nitty-gritty of the framework. But to get the feel of it. Okay, so these are the elements of the draft proposal. We would propose to enforce registration and listing with an option for notification. The main goal here is to get information about the LDTs that are on the market. Right now, we have no systematic way of getting information about these LDTs, and we know there are a lot of them. We don't know what they're used for. We don't know the risks associated with them. That is a mechanism to get that information that's lower burden than making everyone on the market come in and register and list, as their medical device IVD counterparts do. We believe that it's important to enforce the adverse event reporting requirements, because this is a good way, again, to get at that risk question of how often are these tests failing, how can we, if we have this information that will help us in our assessment of the actual on-the-ground risks of these tests. We plan to use a public process, so not only advisory committee meetings that will be publicly informed, but development of draft guidance on the risks and on the prioritization of how these tests are brought into the agency. We would propose to phase in the enforcement of pre-market review and quality system requirements over a 9-year period, a very long period of time, for the high and the moderate risk tests. And I think importantly, we've proposed to continue some enforcement discretion for certain categories of tests. Law enforcement LDTs and certain LDTs for transplantation are completely, would be completely outside of the framework, so they would continue on as they are with complete enforcement discretion generally. So the 4 categories that we believe would be under this general continued enforcement discretion for pre-market review and quality systems. Again, they would have to notify, and they would have to report adverse events, but they would be under enforcement discretion for pre-market review and quality systems, or the low-risk LDTs, so that class 1, where the risks are fairly low in that group. LDTs for rare diseases, we recognize that these tests may not come to market otherwise. There's no business model for them. Traditional LDTs, the type of LDT that was there in 1976, so using FDA-regulated components, a heavy professional component to the interpretation, but no automated interpretation. And LDTs for unmet needs, where you have no counterpart to an FDA-approved or cleared device. And this is a recognition that labs do play a role in innovation, and we don't want to undermine that role of labs. But there should still be some controls on that. So each of these categories, and this is where we can have, if we have time for discussion, I'm happy to get into it, but we are asking for comment on these categories. Did we get the definition of these categories right? Do you see loopholes? Do you see whole things that we've missed? And to really provide some feedback on how these categories were designed. Okay, so the phase-in of the big part of it, the pre-market review, who needs to come into the agency with a submission that we would review it before it could go on the market, or continue to be on the market, as the case may be. So the highest risk LDTs would have to come in first. I don't want to spend time on the actual timeline of this. You can look at it in the document, or I'm happy to talk about it later. But each of these is given a little bit of time to come into compliance, and then once the submission is made, they're still under enforcement discretion until a decision is made by the agency. So there really was thought put into allowing time to come into compliance and not disrupting the market. Highest risk LDTs are considered those that have the same intended use as a companion diagnostic. Very important for personalized medicine. We feel that that's one of the first categories we would want to see pre-market review for. Same intended use as any class 3 medical device, and then certain LDTs for determining blood or safety and effectiveness of blood or blood products. So those are sort of the ones we would start with immediately. After those go through, we will learn through the notification process about all of the rest of the world of LDTs, and we will use a very public process to classify them. These seem like class 3 claims. These seem like class 2 claims. We will engage the community for that effort, put out guidance documents, and prioritize them. So the ones that follow over years 2 through 9 will know when they should come in, and that will be done in a public process with all the high-risk ones coming in and then moving on to the moderate-risk ones. So that was probably too broad, and, but hopefully you can take a look at the draft guidance. We really do want the input. After the period closes, it closes in February, there will be a public meeting and public comments will be taken at that time. So, FDA is not going to publish a draft guidance on February 3rd after the public comment ends. I can guarantee you we will consider your comments and try to make the best framework possible. There is a website that we actually made simple so people can find it. It's not buried, and I would encourage you to take a look at that. Move on to
Good morning. I'm from the New York State Department of Health, and I want to give you an explanation and an overview of our program so that you have a better understanding. So the public health laboratories of New York State are located in the Wadsworth Center. The program that I work for is the Clinical Laboratory Evaluation Program. We are located in the Division of Laboratory Quality Certification. That division is responsible for all of the regulation of laboratories that perform testing on specimens collected in New York. So that includes the clinical program, it includes the physician office program, blood and tissue resources program, breath alcohol that certifies all the police officers in New York that perform breath alcohol by breath alcohol testing devices, the environmental program, laboratory investigative unit that does a lot related to fraud and billing abuse. Our public health laboratories are shown here in this picture. There's 4 different buildings. We do everything from newborn screening to infectious diseases. We are also responsible for regulated medical waste, which is very important right now with the Ebola concerns. The way that New York State public health law is written, it is very broad. And what it says is if a specimen is collected in New York State, that laboratory, regardless of where it's located, must meet New York State requirements, hold a New York State laboratory permit before it can perform that testing on a specimen from New York. So to give you an understanding, we have about 1,000 clinical laboratories, about 5,000 limited service laboratories. Those are the wave testing sites, very low categorization of complexity of testing. And we also certify about 3,500 laboratory directors. So in terms of California and the West, we have a surveyor out here on the West almost every single week. With your startup laboratories here, this is a very big place for technology. It's a big place for genetic testing, for molecular oncology. And up until about the last 6 months, the packages coming in for new permit applications were primarily genetic testing and oncology molecular, but clinical toxicology and pain management is now really what the future is occurring. This is our regulatory authority under public health law, just so you see this. And basically what we do is we regulate everything related to clinical laboratories, including the review of validation. And we look at both analytical and clinical validity. We do not look at clinical utility. That is really up to the insurance companies to decide whether or not they'll reimburse. This is an idea of what we look at. So our review process for validation began in the mid-1990s. We did not anticipate that LDTs would explode the way they do. So far, we have received more than 10,000 validation packages from 565 laboratories, and you'll see this information in the October 3rd publication from the FDA. And some of these include the pure LDTs that are described in the FDA document. Some of these are LDTs using ASRs. Some are RUOs, IUOs. Many are modifications to FDA-cleared approved exempt tests. And then there are many modifications to RUOs and IUO labeled tests. I wanted to give you, because I'm a visual learner, just an idea of what the validation reviews look like over time. So CC is cytogenetics, or clinical chemistry. CY is cytogenetics. DI, diagnostic immunology. GE, genetics. MI is all the microcategories, so bacteriology, mycology, mycobac, virology. ON is oncology. OT is other. And TX is tox. In that other category, you'll see things like endocrinology, cellular immunology, in which almost every test is laboratory developed, cytokines, transplant monitoring, histocompatibility. Our categories are different than CMS's categories. Also, I wanted to mention to you that New York State is one of 2 states that is exempt from CLIA requirements, and that's because we've demonstrated to CLIA that our requirements are at least as stringent as CLIA's, if not more so. The other state is Washington State. California used to be CLIA exempt several years ago. My understanding is they are revisiting that and thinking about it once again. So for laboratories located in New York, we are the primary accrediting agency. CMS goes in there and looks at those laboratories for what they call validation surveys. What those are is they go in after our surveyors have been there to do a quality assurance check to make sure that we're identifying everything that we need to see. Now you can see the subtle changes in validation. You can see a spike here in genetic testing that's related to Sanger sequencing. The things to consider about validation review. So from New York State, we will only approve a validation package that's both analytical and clinically valid, that their performance characteristics have been established and support the clinical claims from the laboratory. Now, that's a big issue. The laboratories do not always submit documentation that supports their clinical claims on that population or using that specimen type. And then all technical procedures employed in a laboratory shall be of proven reliability and generally accepted by the leading authorities in the specialties of laboratory medicine and or approved by the department. So we've seen this also as well in several different areas. Particularly, you'll see this in Lyme disease, you'll see this in some of the other areas. Now, validation review is not required for a series of different things. So standard methods, bacteriology culture is an example, and identification. That does not require validation review. And then we have something called an exemption. If a laboratory submits 3, 5, 6 packages to us for the same methodology, if the laboratory submits a very strong package that demonstrates clinical and analytical validity, we may give that laboratory an exemption and say, for this methodology, we do not need to see additional validation packages. So there are, for a good example, tox laboratories that may perform, 10,000 different toxicology tests. We'll do a sampling of their validation packages, we'll choose what they are, and if they perform well, we may give them an exemption. And then we do not review legacy assays, those that were established in the laboratory prior to April 24th, 2003. In terms of the general requirements for assay validation, it's the analytical performance described earlier, specifications for accuracy, precision, reportable range, reference intervals, analytic sensitivity and specificity, interference substances. We also look for the laboratory to establish applicable performance characteristics, including clinical sensitivity, specificity of novel assays without comparable methods. We look for the laboratory to assure established reference interval that's appropriate for the patient population for that laboratory. And we require that they submit validation data and SOP for review in accordance with our submission guidelines. So some of our categories, such as genetic testing, has very specific information on our website regarding what kind of data needs to be submitted by the laboratory. When our technical reviewers, our scientific staffs, the MDs, PhDs that do this review, it is very rare that a laboratory that submits validation data gets approval on the first throughput. Usually our scientific staff have questions. It goes back to the laboratory. They have a specific time to answer those questions, or the package will get inactivated. So when the FDA, for example, will ask us questions about how many packages have we denied, that's a difficult question because you've got the outright denials, and then you've got the letters that you send to the laboratory saying analytical validity is missing, clinical validity is missing, these are the things that are missing in your package, this is what we expect to see, these are the questions that we have, and they may choose not to respond, in which case we'll inactivate the package. It's truly a form of a denial, but it's much more difficult to quantify. So in terms of our goal is really about the accuracy and reliability of the test result. We have received many packages that are either analytically fundamentally flawed, clinically flawed, or they don't support the claims that the laboratory is saying that they're looking for. So now in genetic testing, we're seeing some very new packages come in, which would at this point be considered low risk by the FDA, weight management, fitness, those type of things. But then we're also seeing much more complicated things related to things like fibromyalgia, which is a very difficult concept when it's a rule-out disease, or suicide risk packages, depression packages. These are much more complex, much more difficult to look at in terms of clinical validity. You know, the analytic validity, as described earlier, is much easier to get that down, but a clinical validity for a fibromyalgia package, that's a challenge. Thank you.
I think I need to make all the speakers Hank Greeley. Again, I'm going to assert a privilege whether I've got it or not and ask the first couple of questions, but then questions at the microphones at both sides. First one, Kathy. Really interesting what you guys have done in the genetic testing realm as a non-regulatory agency, as a research entity, between the genetic testing registry, ClinVar, GenVar. It seems as though, and perhaps this is because both you and Dr. Collins come out of a genetics background, you've been, in the relative absence of substantial federal regulation, you've been setting up almost a shadow system or a voluntary system. How do you see that changing if FDA regulation becomes more significant? How are you two going to work and play together?
So I think the resources that we try to create are really in the spirit of data sharing and enablement. So we now, and I didn't mention, we have a new policy about genome data sharing, which requires our grantees, if you're generating genome data, whether it be from humans or from critters, that you have to have a data sharing plan, you have to deposit it into a resource. So we are big on data sharing and building resources. One of the things I will say, and we have been in ongoing conversations with FDA through the development of their guidance and subsequent to it becoming public, about how we can have our resources and our expertise be helpful. And one of those is exploring the notion of how potentially the notification, which is actually really clever, right? So FDA came up with a really clever way of saving laboratories money, but still collecting the information that they need through this notification rather than registration and listing process. So you could potentially see notification being through submission through the genetic testing registry. You could also see some CLIA functions being accomplished through the genetic testing registry. So to the extent that that can be a useful portal, we would like to be able to make that helpful. In terms of assessing clinical validity, I think we do have the kinds of expertise that FDA will be looking for, and we've been talking about how we can provide advice. They are the regulators, they're the deciders, but we can provide inputs to them.
Okay. And then a similar question to both Stephanie and Pamela. As we move, if to the extent that the federal government takes a stronger regulatory stance with respect to these kinds of tests, what's the state-federal interaction like?
So we work quite closely with both CMS and the FDA. And I think at this point we've had a very good working relationship with both. So CLIA, the CMS people, do not look at clinical validity. They take basically a very hands-off approach to that concept. They're looking at analytical validity. They're looking to see that the laboratory has done some type of validation study, but they're not looking at that. So we've been discussing with FDA for a very long time about what's involved with the validation review, what do we do, and we regularly share data with the FDA when they're trying to look at, how to do this. I mean, the information that we provide, and I want to make this a point, is that we do not know the denominator for the number of laboratory-developed tests that are out there. I mean, certainly we can tell you what we've seen so far, and that's just specimens that are tested that were collected in New York, and that's over 10,000. Well, every single state is developing laboratory-developed tests. That is the way the laboratory field has gone. So the denominator is large.
Yeah, I will just reiterate that FDA does not want to reinvent the wheel here. So to the extent we can leverage activities that are ongoing that meet that regulatory bar that we have to meet, we are happy to continue to engage.
Although one of the interesting things is, in terms of reinventing the wheel, New York has a very fancy, very well worked out, regularly used wheel. Not every state has that. Some of the states have square wheels, I think, with respect to this kind of testing. And so FDA does, encompasses all the states, the ones with very active and very excellent programs, and the ones with programs that are less developed. And how both the outliers, the best and the most developed and the least developed, will work under a federal context will be interesting to see, I think. Everyone agrees it's interesting.
This is very interesting. I think to some extent New York State has stepped in where there was a lack of oversight. Yeah. And so if there were more comprehensive oversight from the federal level, some of those outlier at the bottom would not need to step in the same way that New York State has. So.
It's sort of NIH has stepped in into a vacuum, not with regulation, but with information, but you can't do regulation without information. Okay, let's take some questions.
Yeah, I had a question for Sherman and Dr. Bradley, and something that interests me a lot, and that is the issue of when the regulation of a test for clinical validity goes from being a parameter of the test to becoming an issue of medical practice. So a pathologist looks down a microscope and interprets an image, or a radiologist looks at an MRI scan and interprets those pixels. There's clinical validity issues involved in what they're doing. So are they regulated in the same way, or is it a legacy issue because H&E staining goes back 200 years and radiology goes back 100 years? Where do you draw the line between parameter of a test, when you're talking about clinical validity, and interpretation of a test, which is medical practice?
Do you want to start?
It's a really good question. So there's a lot of scope of practice issues. And do we look at scope of practice? Do we regulate pathology stains, et cetera? Yes, we do. And do we look at validation packages related to that? Yes, we do. And then we also, in those validation packages, look for interpretation. And we're looking at all of that interpretation, which of course are coming from the pathologists.
Yeah, I sort of thought of the H&E being more of an analytical validity question rather than a clinical validity one. Similarly, you regulate an MRI scanner, should generate the right pixels, but who interprets those pixels and what the image means is another story.
Right. It gets into bioinformatics.
Right. And I think this is one question that we'll grapple with, and we want input from the community on this exact issue, where the test sort of ends and the practice of medicine begins. If you think about something that could be automated, if the result at the end of the day is a piece of software that will take whatever the instrument pops out and gives you an interpretation, that's clearly a device. But what if it's a person sitting there giving you that? So I think good questions and things that we will continue to engage on as we help you understand where in our regulation the device begins and ends.
Thanks.
Yeah, I want to go back to the discussion we were just having between New York State and FDA. So Stephanie, you've gone out there ahead of the FDA, actually. You've sort of paved the way. And Pamela said they don't want to reinvent the wheel. What was your response when you read the draft guidance?
We're very pleased.
You're very pleased. So at the end of your talk, you started getting into, there's some complex tests out there. Are there 2 or 3 things that came to mind or come to mind?
Well, certainly we don't have a complexity model. We look at all, and we have concerns related to all testing. FDA is really looking at those at high risk as a starting point. It's a little concerning to us in some respects, the timelines to put everything in place, but it's an enormous undertaking that the FDA is putting into place. It's not something that can be done fast, because then it would not be able to be done well. But I think that at this point, it's the length of time to put everything in place that we're looking at most closely.
Okay, thanks. Can I ask one more question?
Sure.
Pamela, what about international companies offering tests here? How will you look at that?
Well, if they are going to be doing returning test results to patients in the U.S., then they fall under the U.S. CLIA guidelines, and then the CLIA guidelines would make them fall under the FDA to whatever extent they would.
So are there any complications there in enforcing that?
No. So right now, we regulate anywhere that a New York specimen is tested. That laboratory must hold a New York State permit. But first, they're issued a CLIA number, and they're either regulated by CAP, the College of American Pathologists, or they're regulated by CMS. And because they're performing testing performed on specimens in the United States, the regulatory authority is fully established with those
Power of the
laboratories.
state has been reasserted here, thanks to the microphone being fixed.
So no, there's no problem in regulatory oversight of those sites.
Well, let me push on that for a second. There's no problem in terms of what you want to do, but how easy is it for you to figure out if somebody has mailed a tube containing saliva or some other sample to, say, Ireland or the Bahamas and gotten the results back through the internet? I mean, is there a practical enforcement problem, both at the state level and ultimately at the federal level?
So it's complicated in multiple structures. So we have several laboratories that are located out of the U.S. And we regulate them like other laboratories. If we receive a complaint related to those laboratories, we have to go on site. And in terms of direct-to-consumer, you're really reliant on our laboratory investigative unit that does routine searches to try to find these places, or you're really required to rely on the complaint investigation. Otherwise, it is certainly the possibility that these things could exist and you wouldn't know about it.
Thank you for a very informative panel. I was excited recently to learn that Wikidata Wikibase has listed all genes. It's a new interlingual database that Wikipedia has created. In developing MIT OpenCourseware-centric World University and School, as a startup university, we would like to be in all languages and countries and have a genetics aspect and also have a research aspect, potentially have a counseling aspect, and a series of regulatory questions. Following up on the international questions that have just been addressed, are you engaging in any way in helping to define regulatory process in other countries, for example? And could you see that potential with, say, such an international university that was STEM-centric and in all countries and all languages?
I think certainly we have a lot of interaction with other regulatory bodies. A lot in the drug area. There is less so in the diagnostic area. I think it's important and not out of the realm of possibility, but nothing that I could talk about now.
Jake.
So I have a legal question, I think mainly for Stephanie. I guess I have a question as to exactly what the constitutional hook is to require that any samples that originate from New York have to be certified under New York State guidelines, even if they're in another state. I was wondering if you could speak to that just a little bit. Have you received any pushback from other companies that are outside of the state of New York? And I can certainly imagine that being the case with a number of companies here on the West Coast. And then one other thing to think about is to the extent that there's a laboratory, let's say in state A, that does the actual testing, but there is an outfit that returns results in state B, how exactly does New York State go about thinking about that?
So public health law is very firm, and it describes right in the law itself, in the statute, tested in New York or the specimen coming from New York requires a permit. So there have been laboratories that have challenged that and lost because it's very clear in the statute what that specimen requires in order to be tested. So that has been tried, and the statute, like I said, it's a very strong statute in the way that it's written. We see distributive models all the time. So we're seeing two-part distributive models, meaning that the analysis is performed at one, interpretation, reporting done at the other. But now we're beginning to see 3 laboratory distributive models. So analysis at one, interpretation at another, reporting at the third. We require all 3 to have laboratory permits, and the validation packages submitted must be validated for all 3 laboratories to perform that component.
So NIH's mission is to push research forward, and FDA has to provide meaningful regulation. But how, with genetics moving forward so quickly and constantly being new generations and iterations of things, how does FDA become more nimble? Do you work with NIH to do that? or?
I think this is a challenge because things are moving so quickly and new technologies come before us. We are continually trying to bring in the expertise in-house, so we have lots of medical officers and people with PhDs in genetics and biostatisticians that are ready. So we are trying to ready ourselves for the technologies that come forward. We are reliant on the technologies coming in with submissions, so we can't force anyone to bring in their new technologies. But when they come in, we try to be ready. We also, I think, have a fair amount of flexibility, as we showed with the next-gen sequencing clearance last year, that we can not require that every single site that you're going to look at has analytical validation for it, but to look at representative samples. So I think we are really pushing the envelope and figuring out ways to do it as they come forward. We'll just continue to work in that way.
The other thing that FDA has and NIH does as well is the ability to call on experts to come in and help. So they have the ability to have advisory committees inform and provide advice to them, as does the NIH. And so it's not like you're limited. Exactly. FDA is limited to its own staff, but can tap into the broad intellectual capital of the country.
So here at the Center for Law and Biosciences, we're aware of biology and biological constraints, which means there will be a break very soon. Let's just go to...
Actually, let me just pile on the answer that FDA...
Let's go to Mark.
FDA has been a terrific support of the work we're doing in developing a framework for analytical validity. And so there's evidence that they not only bring in experts and partners, but they've been terrific support, as has the rest of the community. And I'll give it to Elaine and let Elaine remark about the partnership with AMP.
And so I think you two were there before you were, so we'll just do two last questions, but we will have a break, and you'll have a chance to ask Pam.
Okay, I'm Elaine Lyon, University of Utah IREP Laboratories, but also representing the Association for Molecular Pathology. And I told Mark that he could...
And on our next panel.
I told Mark he could take this time to thank the FDA if he also recognized that the laboratory community in general is also supporting NIST. My question is for Pamela. If you could expound a little bit more on the FDA's thinking of why transplantation is still going to be under discretion. Enforcement discretion.
So if you read in the draft guidance, and I am not going to claim to have it verbatim, there is an explanation in there for why we think that's important to leave it under enforcement discretion. It has to do with how unique each of those tests is at the site of where they're being done, and there's more explanation in the guidance that I'll let you read through.
Hi, Gail Vance. I'm from Indiana University, but also representing CAP at this meeting. Two questions, and they're actually follow-up questions to questions that were already posed. The last one being nimbleness. Stephanie, actually these two questions are for you. Is the public health law that you quote, is that a state law?
It is a state law. New York
so
State public
law
health
is
law.
held up, but I think the question was constitutional law. So have you ever been challenged at a federal level?
I'm not sure that we've been challenged at the federal level. We've been challenged at the, well, actually, no, I believe we have been challenged at the federal level because it was an out-of-state laboratory that challenged.
Okay, and it held.
Mm-hmm.
Okay, the other, now going back to the nimbleness, in your graph you showed the dramatic increases of testing that was done, and in Kathy's presentation there were 23,000 tests that had already registered in ClinVer, or GTR rather. Let me ask you this. For New York State, what kind of resources have you had to mount to enable your staff to review all these packages? And can you also give us an average timeline for a laboratory? And you've already explained that, if the package is not complete, it's held up. But average timeline for approval?
In terms of our personnel, like all states, we're under budgetary constraints. Our staff are not just assigned to validation review, so they're doing research, they're running the clinical laboratories, they're doing all kinds of different tasks in addition to the validation review. So in terms of timelines to get a permit, so there's two different questions that are involved in what you're asking me. So the first question you're asking is, how long does it take to review validation? The second question is, how long does it take to get a permit? So if you were to apply for a permit right now in genetic testing and you were located in this area, and the permit application was complete, meaning you had a director who held a certificate of qualification in genetics, everything was included in the package, I would tell you that we will not be on site to survey you probably for 6 to 8 months.
Okay, so assuming that I have a permit, so that's 6 to 8 months if I bring up a laboratory, right? And
So if you have a permit in the category of genetic testing already and you submit a new validation, you get something called conditional approval, and you may begin testing in that category right away. And we will review that package. If there's any problems, we will get back to you. It's really, the question that you're asking me is related to an initial laboratory that has not got a permit yet, and how long will it take before that initial laboratory gets a permit? Well, it depends on the permit category. It depends on the quality of the validation package. It can be anywhere from 9 months to longer.
Okay. And is there a fee associated with this?
The application for a New York State permit is $1,100, and you are responsible, you being the clinical laboratory, is responsible for paying our surveyor costs to come to your site to survey that laboratory.
Okay, thank you.
Okay. Well, join me in thanking this panel.